Right, so we ended up last week with this slide to, I hope, illustrate why bacteria perform fermentation. And our objective today, well, before we go on to other types of energy metabolism, is to look in a little bit more detail at some specific fermentation pathways. So I think this is, you may not realize it, but this is in fact a simplified overview of some fermentation pathways. There are many others, and this is just some of them that are illustrated. So they all start from pyruvate here, and they're producing other things, a variety of things: ethanol, lactate, formate, butanediol, propionic acid up here. And so every time I get some kind of diagram like this, has got arrows going everywhere, I always think, now, why on earth does it have to be so complicated? Right? So that's my main objective here, is to try and explain why different types of bacteria do different things. They don't all use the same fermentation pathway. And that's because some of the simpler ones have big disadvantages. Okay? So they don't perform these complicated pathways just to make our lives a misery, well, your lives a misery as students. They're doing this for a reason. So overall, okay, there are several different pathways. Pyruvate is always the electron acceptor. You know, it's ultimately all coming from pyruvate. This is where the electrons are going to go somehow. So if this comes up as a question in an exam or test, all right, during lactic acid fermentation, what is the electron acceptor? Pyruvate, okay? And if pyruvate is not the direct electron acceptor, write something about in your answer. It's something else derived from pyruvate. So ultimately, pyruvate is the electron acceptor. And then it will be difficult for anybody to say your answer is wrong in that case. Okay, so let's have a look at some simple fermentations. And for each of these, it's always good for you to know one or two specific examples, okay? So this is also a very easy question for us to ask you in tests. Give an example of fermentation and what bacteria use that pathway. Or give two different fermentation pathways, cite the bacteria that use those pathways. Okay, so learn a couple. So lactic acid fermentation. This is really simple. Okay, you have pyruvate. It's the end product of glycolysis, and it's going to be used directly as the electron acceptor by lactate dehydrogenase. So 2 electrons from NADH are going to be added onto pyruvate. This will produce lactate. So it's this group here is going to be reduced to a hydroxyl group. So many bacteria perform this one. Lactobacillus. So Lactobacillus bulgaricus in yogurt, Streptococcus thermophilus in yogurt production. Lactococcus also does this. And as I said, fermentation can be homo or heterolactic. Homolactic fermentation is Streptococcus thermophilus. When you make yogurt, they only produce lactic acid. Okay, they don't produce any ethanol. Actually, that might be an interesting idea for a product, right? Yogurt that gets you drunk. And okay, also another simple one, ethanolic fermentation. So first, pyruvate is going to be decarboxylated. So you get rid of this carboxylate group here. This is kicked out as a carbon dioxide. That leaves you, what do you call this? Acetaldehyde, right? I believe so. If not, you'll have to check an organic chemistry textbook. And then this will be reduced to ethanol. Okay, so this aldehyde group here is going to be reduced to a hydroxyl group. This gives you ethanol. So that's of course performed by yeast mainly, Saccharomyces cerevisiae, to produce wine, beer, bread. Okay, CO2 for producing the gas in the bread and alcohol. Since actually that saying look things up in an organic chemistry textbook reminded me a little bit of something I said yesterday was not quite right. So if you remember, I was talking about different nucleotide triphosphates and their uses in synthesis of polymers. So UTP, that's correct. I said polysaccharide synthesis. CTP, this is for lipids. And GTP is directly involved in the peptide bond synthesis by ribosomes. This is the energy source for the peptide bond. ATP is used for amino acyl tRNA synthesis, charging the amino acids onto transfer RNA. So what I said last week was slightly correct. I couldn't quite remember the way round it was, and it's this way, and this is the correct use of nucleotide triphosphates. Okay, so these two simple pathways, just one step or two enzymatic steps here for ethanolic fermentation, and that's it. You get rid of the extra electrons, which is the whole point of the fermentation, so that this NAD+ is available to be used in glycolysis so you can produce more ATP. All right? It's done very, very simply. Why don't all bacteria do this? Why do they do something more complicated? Well, the main drawback is that the fermentation products are toxic or can be toxic when they accumulate. So lactic acid is going to reduce the pH, which will inhibit bacterial growth as pH goes down. Ethanol. You know, why can you only ferment wine up to about 14 or 15%? In the end, the yeast get killed. It's toxic. So that's the drawback of these simple fermentation pathways. They produce toxic end products. Now, some of the slightly more complicated pathways are a slightly better solution in terms of lowering toxicity. So one example is butanediol fermentation. This is very, very common among bacteria. Okay, Serratia, I remember this one personally, Enterobacter bacillus. So gram-positives and gram-negatives can do this. Once again, you start off with pyruvate, but you're not going to reduce it directly. So one thing you have to get right here, this is 2 pyruvate molecules, right? So you start off with 6 carbons. You kind of stick them together, you lose the CO2. So this, you have 5 carbons here. You kick out another CO2, so you've got 4 carbons here. And then this step, this is the step where you're going to reduce, once again, an aldehyde group. Well, you might call it a ketone group because it's 2,3 really. It's not on the terminal carbon. And this will give you to a hydroxyl group, and that gives you butanediol. So the advantage is that this is a slightly less toxic end product. Yeah, another thing I didn't mention explicitly, but of course, okay, lactate fermentation, ethanolic fermentation are very important in the food industry. This one used to be used in industry because butanediol is a starter compound for the synthesis of synthetic rubber. But ever since the 1950s, the bacterial fermentation has been replaced by just petrochemistry. So it's not used industrially anymore, but it used to be important. Okay, so that those fermentation pathways are quite clear. Okay, for lactic acid and ethanolic fermentation, you should know the steps, okay? Because it's one step or two step. You should know that. But for this one, I don't think you really need to know every single step. And in the other ones as well, you won't need to know exactly what the different intermediates are. Just try and remember what the end product is, which bacteria do it, and kind of have an idea of why they're doing things like that. That's the objective. Now for the other ones, they look complicated, but they aren't really, because what bacteria are doing are using enzymes that they already have that are involved in other metabolic processes. And in order to be able to understand that, you have to know a little bit about what are the other metabolic uses of pyruvate. Actually, it's really at the center of a lot of metabolic pathways. So, for example, pyruvate can be carboxylated. This gives you oxaloacetic acid. And that's at the beginning of many pathways of amino acid biosynthesis. Another main use of pyruvic acid is to produce acetyl coenzyme A on the basis of lipid synthesis. It's going to start from here. And in bacteria which are aerobic or facultative anaerobes, acetyl-CoA derived from pyruvate is going to go into the tricarboxylic acid cycle, also known as the Krebs cycle. Now you do this, I believe, in the second semester, but you have to, I think, right now have a little bit of an idea of what is going on. So just count the carbons, okay? This is 4 carbon here. This is 3 carbon. This is 2 carbon. So these two are combined, and the first thing you get is citric acid, which is 6 carbon. Converted to isocitric acid and to alpha ketoglutaric acid. This is 5 carbon, so CO2 is coming out here. Succinate, fumarate, and malate. And finally, this will be, so this again is 4 carbons here, so you get another CO2 coming out here. So finally you get, this is all recycled round to oxaloacetate. What has happened here? This is an acetyl group. So basically you start off with 2 reduced carbon groups on coenzyme A. And you kick out carbon dioxide at 2 steps. And the electrons that are here associated with this acetyl group, they produce NADH. And if you're an oxygenic bacterium, you respire, and then these electrons go on to oxygen. Oh, a little bit of a question here. Malate, oxaloacetate, where have we seen these before? Last week? Bacterial nutrition and import pathways. These are the examples of carbon compounds that are imported by. 3 choices: group transport, ATP binding cassette, or proton symport. It's this proton symport, okay? So these are acids, oxaloacetate. That's why they can come in with proton symport. Okay, that's just a little reminder. Okay, so that's what pyruvate is going to be doing in a bacterium that can respire in the presence of oxygen. Now, imagine you are a facultative anaerobe. Okay, in the presence of oxygen, you're going to be doing this. You're going to be performing the TCA cycle. getting rid of all the carbon here. It's going to be completely oxidized to CO2, and it's expelled from the cell. You get a lot of NADH, and you use that to make ATP because the electrons go onto oxygen. Now, imagine you're stuck. You use up all the oxygen, you're down at the bottom of the lake or something, or there's no more oxygen in the test tube where you're growing, but you still have all of these enzymes that are around. So in order to convert this respiratory metabolism into fermentation, you can use some of these Krebs cycle enzymes in the other way around. You run them in reverse. So pyruvate to oxaloacetate. Okay, we saw that, so that's where oxaloacetate comes from. And then all of these enzymes here, this is the Krebs cycle in reverse. So you've already got these enzymes. They're the same things. They're just running the other way around. And then at the end, you're going to kick out CO2 and excrete propionic acid. So this is propionic acid fermentation. Okay, very easy one to remember. Propionibacterium performs this. And the last one that you have to worry about is mixed acid fermentations that are performed by a lot of coliform bacteria. The enterobacteria do perform this. Salmonella, Escherichia. So, okay, mixed. Okay, remember the examples. Okay, lactic acid fermentation, Lactococcus, Lactobacillus, ethanolic fermentation, Zymomonas, butanediol, Serratia. Propionic acid, propionibacterium. Mixed acid, Escherichia coli is the one you want to remember. Okay, so what is happening here? Okay, it all starts from pyruvate again. And as we saw just now, one of the main metabolic steps from pyruvate is to form acetyl coenzyme A. Now, normally, when this is done as part of aerobic metabolism, then it's performed by pyruvate decarboxylase. Carbon dioxide is kicked out, and you get formation of another NADH. That's exactly what you don't want in a fermentation reaction, because you're trying to get rid of the electrons. You already have too much NADH. You're trying to recycle things to recycle this to NAD+, to the oxidized form. So in a mixed acid fermentation, pyruvate is converted to acetyl coenzyme A using a different enzyme. Okay, so this is called pyruvate formate lyase. So instead of carbon dioxide being liberated, you get formic acid, formate. And so that means that the electrons stay here on the formic acid, and they're not shifted onto NADH. Okay, so you start with acetyl-CoA, and then so formic acid is the first fermentation product here. Then from the acetyl-CoA, you're going to end up reducing it and getting rid of some of the NADH. By a multiplicity of pathways. Now, really, I don't know this by heart, as you have probably guessed. But one thing to remember about this is that some of the steps, or one thing to notice rather, is that some of the steps allow you to synthesize ATP. So really, the advantage of the mixed acid fermentation is that somehow you can get some more ATP being produced, which is not the case of the other fermentation pathways we've seen so far. So the answer to the question, why are things so damn complicated in this particular case is because it allows you to squeeze out a little bit more ATP production. So mixed acid, you have formate, you have butyrate, you have acetate. Once again, the main problem for bacteria performing this fermentation is the accumulation of toxic fermentation products. So all of these acids reduce the pH of the medium and will inhibit bacterial growth. So one thing that E. coli does is that once the pH goes down, then it will start breaking up this formic acid, producing an enzyme that breaks up formic acid to hydrogen and carbon dioxide. So at alkaline pH, mixed acid fermentation will produce a lot of formic acid. But at acidic pH, then bacteria will start to produce carbon dioxide and hydrogen gas, just to try and prevent the pH from getting any lower, because that would inhibit bacterial growth. Okay, before I go on to that, so let's have a little bit of a question to see if you've understood the basics of fermentation. So I'm going to send out a clicker question. Oh, and I don't have it on a slide, I'm afraid. So I'll slide this over here. Okay, there's the question. Okay, so here's the session key up there. And here are your options. So what are the limitations of fermentation? That's right, that's acetyl coenzyme A, right? Not what I wrote before, I believe. Okay, we have 10 answers, so let's see what you think. Let's have a look here. What have we got? Okay, so what do people say? Okay, this is right. Okay, 8 people out of 10 got this one. End products can be toxic. Acids can turn the pH, lower the pH. Alcohol, it's toxic. So that's a problem. Requires cytochrome. Okay, that's wrong. Only used by gram-positive. Also incorrect. So everybody is right in rejecting these two. And here we got 3 people say energy low per carbon is low, and 3 people say it requires oxygen. This is wrong. Okay, one thing you really have to remember: fermentation is anaerobic. Only occurs in the absence of oxygen. Right? So it doesn't require oxygen. It's totally, completely the opposite. Okay. If you have no oxygen, then fermentation is a possible form of energy metabolism. And this one, okay, this one, energy yield per carbon, so that's also correct. Okay. So you start out with glucose. Remember, you're only getting about 2 ATP per glucose molecule from a fermentation pathway. Maybe you can squeeze out a little bit more if you use mixed acid fermentation. But this is basically low. And you're also kicking out some reduced carbon compounds like lactate, ethanol. These have a calorific nutritional value. That's why we can, eat them. And we can gain some energy value from eating lactic acid in yogurt. Because fermentation is pretty inefficient. Okay? So as long as you have a lot of reduced carbon, well, you can do it. You can scratch a living by fermentation, but it's not great. Okay. Right. So up to now, these are the things you have to really, really know. What we saw last week are the basic nutritional types: chemotroph, phototroph, organotroph, lithotroph, autotroph, heterotroph. Never ever confuse these two terms, okay? Auxotroph, autotroph. Auto, it makes everything itself. It can synthesize. No, that's not what it means, right? It means that it doesn't need organic carbon. It can fix inorganic carbon. Auxotroph, this is a mutant that needs some supplement, needs a vitamin, needs an amino acid, needs a nucleotide, or something like that. In terms of fermentation, okay, what's the problem? Glycolysis produces too much NADPH, not enough ATP. Can you use an organic metabolic intermediate, basically pyruvate as the electron acceptor, and fermentation products can be useful for us, industrially or in the food industry. And it only occurs in the absence of oxygen. Okay, so now we're going to try and understand respiration. And the first way to do that is to look at the kind of respiration that we all know, which is aerobic respiration. Now, there are 3 basic steps here. Okay, the first of them is glycolysis. You start with sugar and you get down to pyruvate. You produce NADH and some ATP. The second step is the Krebs or the tricarboxylic acid cycle. So this pyruvate, as I said before, is going to first be transferred to acetyl coenzyme A, and then these 2 carbons here are going to combine with oxaloacetate and then be completely oxidized to CO2. So if they're completely oxidized, the electrons have to go somewhere else. So they go onto NAD+. So you generate a lot of NADH from the Krebs TCA cycle. And the last step is electron transport and oxidative phosphorylation. So electron transport will be transferring these electrons from NADH onto oxygen, which is the terminal electron acceptor. And during that process, it's going to be possible to generate a proton gradient and use that to produce ATP. So as I said before, I think you do this in biochemistry in the second semester, the Krebs cycle. So I'm not going to talk about these reactions really. All you have to know is that these carbons are completely oxidized to CO2. So CO2 is the excreted product. All the carbons converted to CO2, and you get a lot of NADH. What we're interested in really is how the electrons here are transferred onto oxygen, and how we can use that to produce ATP. So we're going to look at this step, electron transport and oxidative phosphorylation, with our favorite example, which is, of course, Escherichia coli. Now, for the purposes of L2 microbiology, the thing you need to know about electron transport in coli is that there are three main complexes: NADH dehydrogenase, the quinone cycle, which is here, and the cytochrome oxidase complexes. Okay, let's see what's going to happen first. So NAD dehydrogenase. So the electrons start here, and they're shunted first onto a flavoprotein and then to an iron sulfur protein. So FAD, FES goes in alphabetical order, FA first and then FE afterwards. So NADH is recycled back to its oxidized form, NAD+. While this happens, there's a possibility to transfer the protons across the membrane. So this is the cytosol, this is the periplasm. So you transfer the protons here, they don't stay in the cytosol, they get transferred out to the periplasm. The next thing is, okay, so then afterwards these electrons are transferred onto quinone. So this is a membrane soluble molecule. It's hydrophobic, it's in the membrane. And it cycles from the cytosolic phase to the periplasmic phase of the membrane. On the cytosolic phase, it picks up protons. And on the periplasmic phase, it gives them up. So when you have reduced quinone here being reoxidized because the electrons are transferred onto the cytochrome oxidase complex, protons are given up into periplasmic space. Here they're added on here. And the last step is transfer of the electrons in from the different cytochromes in this complex onto oxygen. And this will give you water as the end product, which is absolutely wonderful because CO2 and water are not toxic. So it's a big advantage compared to fermentation already. And also in this last step, at least with what is called the BO cytochrome oxidase complex, then protons can once again be transported from the cytosolic phase to the periplasmic phase of the membrane. So what's the net effect here? The net effect is you've got protons that are being pumped across the membrane at every step during electron transport from NADH, this is where the electrons start, onto oxygen, this is where they end up. So by doing this, you've formed a proton motive force, proton gradient. And this is what can be used to form ATP with the proton-dependent ATPase. So 3 protons will be transported across the membrane, and that allows 1 molecule of ATP to be synthesized from ADP and inorganic phosphate. Now, apparently, if you add up all the protons that can be transported across the membrane from one NADH molecule and the number of NADH molecules that you produce by glycolysis and the Krebs cycle, then there's a maximum theoretical yield of 38 ATP per glucose. So this is at least 10 times higher than what you get from fermentation. This is the jackpot, all right? So one glucose is going to give you 10 times more ATP. Okay, so that's the maximum theoretical yield if you have a lot of oxygen. So in high oxygen conditions, E. coli will use this BO branch of the cytochrome oxidase. But in low oxygenation conditions, then it will switch to using the BD branch. Now the BD cytochrome oxidase has got a higher affinity for oxygen, so it can still work if oxygen is at a low concentration. But it's less efficient in terms of energy production because it doesn't translocate protons across the membrane. So the BO branch does, while the BD branch does not. So BO, okay, O, oxygen. You've got a lot of oxygen. BD doesn't have O, doesn't have oxygen. But the advantage of having this second system is that, you can still carry on respiring actively even if oxygen is starting to get used up. Okay, so that's oxidative respiration, or aerobic respiration. Now, it might come as a surprise that anaerobic respiration is also a possibility for many different bacteria. So with aerobic respiration, okay, this is the electron transport chain that's involved. Okay, you start out with NADH dehydrogenase, quinone cycle in the membrane, and then cytochrome complex. And at the end, you get electrons transferred onto oxygen. And what happens if your oxygen runs out? Well, you can switch to fermentation, but you may find that some other good electron acceptors are still present. For example, if you've got nitrate in the medium, look at these redox potentials here. You've got NADH. The redox potential is minus 0.3 volts. For the half reaction from nitrate to nitrite, the redox potential is plus 0.4. So this is a very, very favorable electron transfer here, okay? Oxygen is down here. Oxygen is always the best, right? But this is still pretty good. So if you've got nitrate, which is present, and you want to get rid of the electrons on NADH, this is a very, very favorable reaction, okay? Okay, much better than like producing acetate or producing lactic acid from pyruvate, okay? So this is what some or many bacteria can do. An example is Pyrococcus denitrificans. So the start of the electron transport chain is the same. NADH dehydrogenase, coenzyme Q in the membrane. What's different is this cytochrome complex. So this cytochrome complex is not going to transfer electrons onto oxygen. Firstly, they're going to be transferred onto nitrate. This will produce nitrite, but nitrite is very toxic. So it's helpful if it can be further oxidized to dinitrogen gas. And that will give you a harmless end product. So once again, this is useful for bacteria because during the electron transport, protons are getting transferred across the membrane, either from the NADH dehydrogenase, from the quinone cycle in the membrane, and also from one of these cytochrome complexes. So once again, if you transfer the electrons onto nitrate, during that process you can form a proton motive force, and you use that to produce ATP. Now, anaerobic respiration can occur with a variety of electron acceptors, because oxygen is not the only possible electron acceptor, nitrate is not the only possible electron acceptor. Every time when you've got a redox potential, which is more positive than the redox potential for NADH being oxidized to NAD+, you can perform this electron transfer. The reaction's going to work. It's going to go in the right direction. So, if you have an underwater aquifer, under the ground, you'll get progressively less and less oxygen because it's going to be used up. You'll have organic carbon at the beginning. This is from vegetation that's been buried or something like that, or bacteria that have been killed by viruses, by bacteriophage. So at the beginning, you've got organic carbon for the beginning of the metabolism. And bacteria will use different inorganic electron acceptors in order to perform anaerobic respiration. So they use the best electron acceptors first, oxygen, then nitrate, then manganese, then iron, and then sulfate. And then at the very end of the chain, carbon dioxide can be used as an electron acceptor for the production of methane. So which bacteria are going to do this? Okay, for nitrate-based anaerobic respiration, you'll have Bacillus, Pseudomonas, Paracoccus can do this. Sulfate being reduced to sulfide, desulfur Vibrio. Okay, carbon dioxide being reduced to methane, methanogens. Don't need to know any names really, they're archaea. I think that Pseudomonas again can use iron. Sometimes you can have weird, arsenates or selenates can be used as a source as electron acceptors, okay, because they've got a lot of oxygen on them. Okay, so try and bear that in mind with anaerobic respiration. We're talking about chemoheterotrophs here. They use organic carbon as a source of electrons and the source of carbon for making cells. And they get energy by transferring the electrons onto an inorganic electron acceptor. Chemolithotrophs are doing practically the opposite. Okay, the source of energy, the source of electrons, is inorganic. And they're going to use the energy they derive to fix carbon. So they only have access to inorganic carbon. So they're trying to do the opposite thing. Now there are three types that we have to know about of chemolithotrophs: hydrogen oxidizers, nitrifying bacteria, and sulfur oxidizers. So what is happening here is that we're looking for some kind of reduced, molecular hydrogen. It's reduced basically, or reduced nitrogen or reduced sulfur compounds. These will be electron donors, and we can produce energy by shifting them onto a good electron acceptor, which is generally going to be oxygen. Okay, so chemolithotrophs need a good electron acceptor. Mostly they need oxygen for their metabolism to work. Not necessarily, but mostly they do. Okay. So they're doing the opposite thing to anaerobic respiration. Why does it work? The big difference is the availability of oxygen. Okay. Here, this is going to work because you don't have any oxygen. If you want to live by chemolithotrophy, you have to have oxygen present, except for the case of hydrogen oxidizers. Okay, so let's have a look at what is happening with hydrogen oxidizers. Well, know the couple of names of hydrogenophaga, eats hydrogen, or Aquifex, makes water. So that's easy ways to remember the metabolism of these two genera. The electron donor is hydrogen, the electron acceptor is oxygen. Massively favorable, it can explode if you it can generate plenty of energy. Now, it's very easy for hydrogen oxidizers to produce both NADH and ATP, because the redox potential of this half reaction, so molecular hydrogen giving up its electrons, is even more negative than the redox potential for NADH to give up its electrons. So that means that NADH can be produced directly. Okay, there's an enzyme, it gets molecular hydrogen, produces NADH because the delta G is going to be favorable here. So that's easy. And while you're doing this, you can, of course, use the NADH later on to transfer electrons onto oxygen, right? That's a terminal electron acceptor. So you have an electron transport chain. You can produce proton motive force. And that way you can get ATP. Okay, so let's try and summarize this. You start off with the electrons that are on hydrogen. They go on to NADH. Then they're going to go on to an electron transport chain. So you're going to have, I don't know, the three things that we talked about. NADH dehydrogenase, quinone cycle, and cytochromes. That will shift the electrons onto oxygen, produce water. That's what's happening. And this is going to produce proton motive force. Then you can get ATP. So that all works because molecular hydrogen is a great electron donor. It's the best. In fact, okay, so another thing that I can say about this is that some hydrogen oxidizers don't need oxygen because they can take electrons from hydrogen and put them onto sulfate. So they live on this reaction, they produce hydrogen sulfide. So there are some anaerobic hydrogen oxidizers. What's the other thing here? Okay, yeah. Methanogens, that's an example, right? They don't need oxygen. Hydrogen is the source of electrons. CO2 is the electron acceptor. Now, where do these two things come from? Okay, think of a biological system that produces methane. Can anybody give me one example? You can find them on a farm. Cows, yes, they produce methane. So this is going on inside the guts of a cow. Where do these two come from? They come from a mixed acid fermentation from gut bacteria in the cow's gut. Because when the pH starts to go down, formic acid is going to be cleaved to hydrogen and CO2. So some of this can be used by methanogens to squeeze out a little bit of energy by producing methane. Of course, inside the gut, it's an anoxic environment. There's no oxygen in your gut, in the lumen of the intestine. Okay, in the epithelial cells, yes, because you have the blood supply, but in the lumen of the intestine, no oxygen. So the bacterial metabolism is based on fermentation and, in some mammals, methanogenesis. So that's using inorganic, using hydrogen as the electron donor, CO2 as the electron acceptor. Okay, so that's one source of the hydrogen for this process. The other source is going to be geothermal. Okay, so you can have white smokers underneath the ocean which produce molecular hydrogen, and then these types of bacteria will be able to use oxygen in the water to get the energy from the hydrogen. Okay, another type of chemolithotroph are nitrifying bacteria. So they are getting their energy from oxidizing nitrite or ammonium using oxygen as the electron acceptor. So once again, where are these things coming from? I don't know. Might be kicked out at the end of fermentation with of decaying organic matter will produce amines. You might have ammonium, which is present, and it can be oxidized. So overall, the electron transfer is very favorable here. So the bacteria can get some energy out of this process. So if we look here, the couple and a half reaction nitrate to nitrite is up here. So this electron transfer is going to work, right? But the problem is that nitrite is not good enough, is not as good an electron donor as NADH. In fact, it's nowhere near good enough. Okay. So how can you produce NADH from oxidizing nitrite or even ammonium? That's a big problem. So this kind of way for metabolism for electron transport to work is not going to produce NADH for these type of bacteria. So if you've got electrons, they start on nitrite, they're going to have to be transferred onto oxygen and give you nitrate. The only thing you can do here is use this electron transfer to get some kind of proton motive force. So during this electron transfer, protons are going to cross the cytoplasmic membrane and produce an excess of protons on the periplasmic side. How can you, okay, so we can use that to make ATP. That's okay, we saw that. Protons, 3 protons go through the proton-dependent ATPase, you get ATP. It can also be used to drive electron transport backwards through NADH dehydrogenase. How on earth is that possible? Because we just started out by saying that this is not a good enough the electron acceptor here for this to work metabolically. This is why you have to remember physical chemistry and the Nernst equation that we saw last week. If you remember, if you've got a proton gradient, then the potential difference is given by the ratio, okay? It's the number of volts of potential difference you have depends on the ratio of the proton, of the charged ions on either side of the membrane, protons in this case. So if you can build up a strong proton gradient, then this will be convertible into a potential difference which is going to be more than 0.3 volts, right? So you can push the electron transfer in the wrong direction because you've got a very, very high or very, very steep proton gradient. Okay, it's not very efficient, right? So you're going to need to transfer a lot of electrons onto oxygen to build up your proton gradient enough to be able to generate some NADH. So you're going to use up a lot of nitrite or a lot of ammonium to produce a little bit of NADH. Why do you want to produce NADH? Because then you can use that to make membranes. You can use it to reduce CO2 to carbohydrate. Remember, these guys are carbon fixing. They don't have organic carbon. They've got to reduce CO2 to organic carbon. That's why they need NADH. Sulfur oxidizers, last group, you'll be glad to hear. Okay, you start off with reduced sulfur compounds, hydrogen sulfide or elemental sulfur. And the electron acceptors can either be nitrate, so this is quite interesting. This one does not necessarily require oxygen, or oxygen, which is perhaps most frequently the case. So Thiobacillus ferrooxidans, good example to remember of a sulfur oxidizer. And it's a problem in old mine workings. So if you have pyrites, so it's going to have like reduced iron and sulfide, which are present in the mine, then bacteria can use these inorganic substrates to gain energy by transferring the electrons onto oxygen. And that's going to produce ferric iron and sulfuric acid. This is what causes acid mine drainage, seepage from old mine workings that are acidic. And you might think it's just a couple of bacteria doing something they shouldn't be doing, but in fact this is responsible for, or in the past it has been responsible for the production of millions of tons of sulfuric acid running off into the Ohio River in America. Now it's useful to know how the biochemistry of these bacteria works, because then you can just solve the problem by blocking off the mine. So there's no access to oxygen anymore and their metabolism cannot work. Okay, once again, how do you produce NADH and how do you produce ATP? Now sulfur oxidizers have a kind of cute way of making ATP, because 2 molecules of sulfite can be combined with 2 molecules of AMP to produce adenosine phosphosulfate. And then this can then be combined with 2 molecules of inorganic phosphate, and you get 2 molecules of ADP. You kick out the sulfate. So you have a kind of direct coupling between the redox reaction and phosphorylation. So they have a simple way to get ATP. That means that these guys can do something even stranger with their energy metabolism, because it means they're going to run everything the other way. Is they're going to have their sulfite to sulfate metabolism, which is going to produce ATP directly. Then the proton ATPase can work in the other direction, produce proton motive force. This proton motive force can be used to push NADH production, and you use that to make carbon fixation, to perform carbon fixation. Okay, before we finish up with phototrophs, I'm going to give you another clicker question, I think, just to see if everybody has really got these questions about electron acceptor, electron donor, which way around is it going to work. Okay, so let's have a little question about that. If I can. So let's try and get this here. Okay, so let's see if you can get this one. I'm going to shift the window over. Okay, so which one of these are going to work as a basis for energy metabolism? Think about it. So here is the session key. Okay, only 6 people dare answer this question. So let's see what let's see what you think. Okay, what answers have we got so far? Whoa. Well, it's an almost entirely random distribution of answers here. Ah, I know it's difficult, this one. It's tricky. Okay, so glucose as electron donor, nitrate as electron acceptor. 2 people said that's correct. You're right. Okay. It's like glucose electron donor, oxygen is the electron acceptor. That's respiration, right? Nitrate, it's anaerobic respiration. That's the process here, okay? Glucose as electron donor, carbon dioxide as electron acceptor. That's wrong, okay? In respiration, you have glucose as the electron donor. CO2 is the product. It's not the electron acceptor. It's not going to get even more oxidized. CO2 is as oxidized as you can get, right? No, that's the wrong way around. If you're an electron acceptor, you're going to get reduced. But the only electron donor that's good enough to work in this system is molecular hydrogen. methanogens. They use molecular hydrogen. Okay. So that might originally come from some kind of fermentation process, but the only is you can't use glucose directly to shove electrons onto CO2. It's not going to work. Okay, nitrite as an electron donor, CO2 as an electron acceptor. Look at these people. 4 out of 6 you said yes, that can work. No, it's not favorable enough. CO2 is no way good enough as an electron acceptor. So nitrite can give up electrons. It would become like what? Nitrate, right? Be further oxidized. What was it? No, it's not good enough. It's not a good enough electron donor. Let's look at this one. Ammonium as an electron donor, sulfate as an electron acceptor. Does that work? Actually, I don't know about that. Molecular hydrogen as electron donor, nitrate as electron acceptor. Yeah, that would work. That would work, okay? Because molecular hydrogen, it's the best electron donor, right? So it's right at the top of the list. Nitrate, electron acceptor, pretty good. Second best after oxygen. So how is this going to work? I think that's not going to work. The other way around will work. Sulfide, hydrogen sulfide, as the electron donor, and nitrate as the electron acceptor. That would work. Okay, so why does it go this way round? Let's go back. Because a lot of people mostly got these wrong, okay? So everybody got this one right somehow. Not many people got this one, and most people got this one wrong. So this is not really being understood correctly. So why are we working? Why does it work in some ways and not other ways? Okay, you have to look at this. Okay. So what do we have? We had one as like nitrite as the electron donor and CO2 as the electron acceptor. That's not going to work, is it? Because CO2 is like all the way up here. You can get electrons from acetate and put them onto nitrate as an electron acceptor, but it's not going to work the other way around. Okay, we don't have all the half reactions, so it's difficult to see. We don't see where sulfate is, for example. What's the other one that people didn't get before I forget? Okay, molecular hydrogen. Okay, so in order to work this out, it'd be good if everybody could go back at these questions, look at the redox potentials of the half reactions, and figure out what's the right way round for this to work. Okay, what's the best electron donor? What's the best electron acceptor? So you've got molecular hydrogen, glucose, they're right at the top. Something like nitrate is down here. Sulfate is going to be somewhere up here. Reduced sulfur, and then the best electron acceptors: nitrate, iron, oxygen. Okay, now that brings us on to our last subject in just under 10 minutes, which is bacterial photosynthesis. So phototrophs, okay, so they are getting their energy from photosynthesis, and they can either be photoautotrophs, they fix carbon, or photoheterotrophs, they use organic carbon. Now, photosynthesis is widely distributed amongst in bacterial phylogeny. There are some photosynthetic archaea, halobacterium, and then other photosynthetic bacteria are scattered all over the place. Right? So these photosynthetic bacteria did not inherit their photosynthetic apparatus from one very ancient ancestor all the way here. It's a product of natural gene transfer. Okay, so one very clever bacteria managed to invent bacteria chlorophyll and the photosynthetic reaction centers, and then they were transferred between different bacterial species and genera, and even families, to give this distribution of photosynthetic bacteria. So chloroflexi, they're the green non-sulfur bacteria, chlorobic green sulfur bacteria. So we'll just have a look at what photosynthesis is doing in the bacterial metabolism in Halobacterium, green sulfur bacteria, purple non-sulfur bacteria, and cyanobacteria. Okay, we're not going to look at everybody. Halobacterium. So this is archaea. They use bacteria rhodopsin as this is basically a light-activated proton pump. So when bacteria rhodopsin absorbs a photon, it will allow proton transport across the membrane. So this will give you a proton motive force. No electron donor, no electron acceptor. The protons just get across the membrane and they can produce ATP. So these guys are basically Halobacterium salinarum is basically it's an aerobic heterotroph. Okay, it can uses organic carbon, performs respiration to get energy. But if oxygen conditions are low, then it will have trouble producing proton gradient. So photosynthesis is used in low oxygen conditions to boost a little bit ATP synthesis. I'm just going to skip this and go to the chase here with green sulfur bacteria. So green sulfur bacteria, they're doing something different. Okay, so here they have a photosynthetic complex based on bacteria chlorophyll with a type 1 reaction center. So this is supposed to be the archaic form amongst photosynthetic eubacteria. So this is what we think was the first type of photosynthetic system in eubacteria. So here there is an electron donor. It's hydrogen sulfide. It's going to give up electrons, and molecular or elemental sulfur is going to be excreted as the waste product. So what is the point of doing this for these bacteria? Well, as we saw before, sulfide is not a good enough electron donor to produce NADH directly. But it can be if the redox potential of this electron is boosted by absorbing energy from light. So this is what's going to happen, is your hydrogen sulfide, your redox potential here, is not high enough to reduce NAD+. So that's why the electron is going to be transferred here into the photosynthetic reaction center. So then it's going to be boosted up in terms of redox potential. And then this electron can be transferred onto NAD+ and form NADH. So that's what this is doing here. The photosynthesis is being added onto metabolism. This is basically a kind of chemolithotroph. It's using inorganic source of electrons to get energy. Photosynthesis is making this more efficient, allowing the bacterium to produce NADH directly. So if reducing hydrogen sulfide is limiting, these guys are going to be at a big growth advantage compared to just basic sulfur oxidizers. And it doesn't need oxygen as an electron acceptor for this to work. Okay? Because the electrons are being transferred onto NAD+, and then they're going to be used to fix carbon. Okay, but you require a fairly decent electron donor. So this is photosynthesis. It's an add-on to chemolithotrophy. Purple non-sulfur bacteria, what are they doing? They're rather different. This time, a type 2 reaction center, and the electrons are transferred in from cytochrome. They're boosted up, and they just go around. They go round and around and around on this little electron transport chain merry-go-round. But the important thing here is you've got quinone somewhere in this cycle. So quinone can pick up protons from one side of the membrane, transfer them to the periplasmic side. That's going to allow bacteria to create proton motive force. And they can use that for ATP generation. So Rhodobacter, example of a purple non-sulfur bacterium. Once again, these guys, they're basically aerobic or this time anaerobic heterotrophs. So they depend on organic carbon. They don't fix carbon, but they live where there's not so much oxygen, so they can't use respiration to generate ATP. So if they have a little bit of light, then they can generate ATP for free, right? You don't need to use oxygen as the electron acceptor and and have an electron transport chain. And at the end, what you have is cyanobacteria. These guys have got type 1 and type 2 reaction centers. And in this case, water is used as the electron donor, which is just a kind of chemical miracle because it allows you to produce molecular oxygen as the waste product. Of course, up to now, oxygen, that's the best electron acceptor, right? But in this case, the redox reaction is being pushed totally in the wrong direction. Okay, this is the one that produces all of our food, and it's at the base of every, well, not every single, but it's at the base of 99.9% of primary production in the world. Okay, so it's either photosynthetic bacteria or plant chloroplasts doing this that produce organic carbon for us to eat. So this is the one that really makes the world livable. And it's also the reason why we didn't end up like Mars, okay? Because molecular oxygen, once it builds up in the atmosphere, can produce an ozone layer, which protects us from UV. Apparently, the reason Mars does not have oceans anymore is because all the water got fried away by UV radiation and releasing molecular hydrogen, which just escaped off into space, and leaving oxygen to react with all the minerals that are on the surface. So we did not end up like the surface of Mars because these bacteria managed to get oxidative photosynthesis and make the surface livable and produce the ozone layer to keep the oceans liquid on the surface. And really the last thing to say about this is, I'm going to skip this, you'll just have to try and remember that. So this is the kind of slide, respiration. You start off with organic carbon, and this is the flow of electrons to produce NADH and NATP. Chemolithotrophs, they start off with electron transport somehow, and it's both going to produce NADH, which is going to be used to fix carbon, and proton motive force, which can produce ATP. And you have to go back and find and try and understand why only some couples are going to work for this. Phototrophs. So it's photosynthesis is added onto one of these two basic carbon energy metabolic pathways. Okay, so thanks for listening. That wraps it up for today.